Okay, so this is section 3.4 of the textbook, and it's about proteins, which are molecules that have diverse structure and functions. And the learning objectives are listed there at the top. The first one is to describe the possible levels of protein structure, explain how motifs and domains contribute to protein structure, and understand the relationship between amino acid sequence, the sequence of the subunits, and their three-dimensional structure. So what are some of the functions that proteins have? They serve to speed up reactions, so they're called enzymes. They're involved in defense. For example, your antibodies are proteins. They're involved in transport, like hemoglobin transporting oxygen to your tissues. For support, so some proteins, like the protein makes of your fingernails and your hair, and even other proteins that help provide structure for your bones, help provide that support. They're involved in motion, like the contractile proteins that are found within muscle cells. They're involved in regulation, in this case, like hormones, like insulin, for example, which helps you control blood sugar. And they're also involved in storage. So proteins are polymers, which are large molecules made of monomers, and the monomers are amino acids. Each chain is going to be considered or called a polypeptide, and the amino acids, again, are the monomers. And this is a general amino acid here, and in general, the amino acid is going to have a carboxyl, which is one of the functional groups you're responsible for knowing, and the other is an amino. And the amino there, amino acids, so the carboxyl is a weak acid. Now the way the amino acids vary is going to be in this R group here. So amino acid, the structure of it are all listed there. I just went over that. So there's the central carbon, which is right here. The amino group, the carboxyl group, which we circled a while ago, can have a single hydrogen, and then the variable R group, this variable R group. So that's that one right there. These are the 20 amino acids that make up our proteins, and keep going forward here. We do, based on their side chains. These side chains give them different properties. For example, all of those here in the first column are going to be nonpolar, which means they're going to be sort of hydrophobic. And then we have polar, uncharged side chains. So the side chains are indicated by this yellow shaded area. The rest of the amino acids is basically the same, except for the R group. But it's those R groups that give these amino acids different properties. So these are going to be polar, and then over here we have some charged amino acids. You're not responsible for knowing all of the exact structural formula of them, but this gives you an idea that the reason they're variable and is because of the side chain. Depending on how they're put together in long polypeptide chains will determine how the protein folds up and then ultimately its function. So how do amino acids join together? Like always, it's going to be a dehydration synthesis reaction. And the bond that's formed is going to be called a peptide bond. So here, this is one amino acid and then another amino acid, and they're going to line up basically in this same orientation. Amino to carboxyl, and then the next one amino to carboxyl. So this hydrogen here from this amino is going to combine with this hydroxide and get removed in the form of water. And then we form what's called a peptide bond, which is right there. And when we have two amino acids joined together, we'll call it a polypeptide. And when we have many, we'll call it when we have two, we'll call it a dipeptide. And when we have many, we'll call it a polypeptide. There are 4 levels of structure when it comes to protein. For example, and these levels determine the structure and function. The first is a primary structure, which is determined by the coding DNA. And this is just simply the sequence that the amino acids are put together in the polypeptide. Then we have a secondary structure, And here you're going to have interacting groups of in the polypeptide backbone here. So this interaction causes a certain amount of folding or bending in the amino acid polypeptide chain. And two general types are the alpha helix and beta pleated sheets. And then we have, if we continue to fold, so we have the initial folds forming the alpha and beta structures. Then we have tertiary structure where we're going to have continued folding of the proteins, like we're folding it back over again on itself. And this here is going to be due to interactions of the side chains of the polypeptide, those R groups that we were talking about. And it's going to be stabilized by a number of forces, the tertiary structure. This is going to be the final level of structure for proteins consisting of only one polypeptide chain. So at this point, it may become a functional protein unless it's a protein that requires more polypeptides. Some functional proteins have 2 or more chains. For example, hemoglobin, which carries oxygen, is made of 4 folded chains. And we're going to refer to that type of structure as quaternary structure. And that's when you're going to have 2 or more polypeptide chains coming together to form the functional protein. So this diagram kind of illustrates that. The first picture at the top is just showing the primary structure mentioned a while ago, and it's just the order of the different amino acids. Each one of these represents each one of those amino acid, another amino acid, another amino acid, another amino acid. And then we have the secondary structure where we have the beta pleated sheets, and then you might recall that a helix means a twisted shape, so there's your alpha helix structure. And then if we get several of these structures to fold upon themselves over again, then we get our tertiary structure where we become the functional protein. And so you can see pleated sheets right here and alpha helices here and here. And then if we're going to have more than one of these tertiary structures coming together to form a functional protein, then that's referred to as quaternary structure. So make sure you're familiar with these 4 levels of structure for proteins. There are some additional characteristics regarding proteins to be aware of. One concept, for example, is the idea of motifs. And here, there's going to be common features in the secondary structure that we see in polypeptides. So we'll get to that secondary structure for the pleated sheets or helical structures. It would be useful in helping us determine or predict the function of an unknown protein. So if we study the protein and we look at a little section, we would say, hey, there's an alpha helix structure there, or a beta pleated sheet here, and a certain pattern going on there. We know what the function is in other proteins, so we might be able to deduce what the function might be in a newly discovered protein by studying those motifs. And then there's domains, and these are going to be functional units within larger structure. Most proteins are going to be made up of multiple domains that perform different parts of the protein's function. So, for example, there might be a domain that helps attach the protein to the surface of a cell or to the cell membrane itself. And then there may be another domain that is actually functioning in reception or receiving other molecules. So you would have different areas or domains of that functional protein that have different functions to the overall workings of that protein. So here's an example here of a couple of motifs. This is a beta-beta motif right here in the structure, and here we have a helix, a turn, and a helix motif. And then over here on the right we see some domains here. So this area might be a certain domain that we know when we see that certain structure there, it has a certain function, and then there's another domain identified, and then all of this in blue here would be considered domain 1, and each one of those domains would have different purposes in regards to the function of the overall protein. Then there's the idea of chaperones. Early on, biologists thought that the newly made proteins just folded spontaneously. So once you put the amino acids together, they folded in a spontaneous way and formed the functional protein. It's later discovered that there are other proteins called chaperone proteins that help fold the protein correctly. If we discover that there are deficiencies in chaperone proteins, They actually have been connected to other diseases. For example, cystic fibrosis. It's a hereditary disorder, and for some individuals, it seems to be that the proteins appear to have the correct amino acid sequence, but are not folding correctly. So that may be due to the chaperone protein and not to a mutation in the gene for making the protein responsible for causing cystic fibrosis. Cystic fibrosis is caused by a protein that's found in the cell membrane, which helps pump chloride ions across the membrane. But we'll get into that later on if you study those areas of physiology. Another thing to know about proteins is that they can become denatured, which is the opposite of nature, right? So in the natural form, the protein would have its normal function. If the protein loses its normal folding pattern, then it loses its function. Structure does, because structure does determine the function. And the things that can cause this denaturation include pH that's outside of the range that's normal for that protein. For example, the protein pepsin in your stomach is only functional at a very low pH because there's an acidic environment in there. And if you lose that the acidity in your stomach, then the protein pepsin will not function properly to help digest proteins. And then here we have temperature. Temperature can cause a misshape. And then the concentration of ions themselves can cause a misshape, because this disrupts the overall interactions within the folded protein, the interactions between the amino acids along the chain. So here is an example here, we just in a diagram form showing, let's say this is a normal shape of your protein, and then pH or temperature, ion concentration changes, and all of a sudden the protein becomes denatured and misshapen. Now, if you reverse the conditions that cause the denaturation, the protein might fold back to its normal structure. However, if you go too far, or say too high a temperature, then you may damage the protein beyond repair. For example, this is what happens, for example, when you cook eggs. The egg white is normally clear, but then you cook it and the protein denatures and it turns to this white solid form. So now we're going to look at our final group. These are lipids, which are hydrophobic molecules. And that means that they are fearing of water. It's because they're nonpolar and water is polar. Water can only dissolve polar and ionic substances, but nonpolar, like lipids, water cannot dissolve them. It's good to remember that like can dissolve like. So in this case, polar can dissolve polar, and nonpolar can dissolve nonpolar. For example, we got two different types of oils, completely different oils, and you put them together, they would dissolve in each other because they're nonpolar and nonpolar. But if you get polar and nonpolar, then they cannot, they're not alike, so they can't interact and dissolve. What are the learning outcomes here for lipids? So we need to describe the structure of specific types of lipids called triglycerides, and then explain how fats function as energy storage molecules, and then apply our knowledge of structure of phospholipids to the formation of membranes. Phospholipids are a modification of a triglyceride, and they help build our cell membranes, or plasma membranes. Okay? So what are lipids? They're a loosely defined group of molecules with one main characteristic. They are hydrophobic, which means they cannot dissolve in water. These lipids are going to have a high proportion of carbon-hydrogen bonds, so that means they're going to have less oxygen. Carbohydrates have carbon and hydrogen, but they also have a significant amount of oxygen as well. Well, here for lipids, you're going to have less oxygen and more hydrogens bonding, and that's kind of one of the differences here. Lipids include fats and oils, which are the triglycerides we're going to describe in a little bit of detail. They also include waxes and some vitamins and steroids, which are like hormones like testosterone and estrogen. So what are triglycerides? These are going to be composed of one glycerol and 3 fatty acids, and I'll show an image of that in a bit. So that's something to remember. Glycerol is a 3-carbon alcohol. And a fatty acid is going to be basically a carboxyl group, which is a functional group joined to a long chain of hydrocarbons, which are hydrophobic. So this is just a chain of carbons with hydrogen attached to them. Now, the fatty acids are not necessarily identical. There's different kinds of fatty acids, and the chains can vary in length. And there's two kinds of fatty acid categories. There is saturated, and then there is unsaturated. For saturated, you have no double bonds between the carbons, and they're going to have a higher melting point. And so what we mean by no double bonds, so if we have a long chain of carbons here, and I'm just going to put a dot there to show there's many more carbons there. It's going to be saturated because each carbon can hold a maximum amount of hydrogens. So it's saturated or full. Remember that each carbon can only hold or can only form 4 bonds. Okay? So if each carbon is joined to the other carbon by a single bond, then we can maximize the number of hydrogens added there. And here, these are typical of fats that have the fatty acids that are saturated, because the chain will lay out straight, and the straighter the chain, the more neatly they can fit together and basically be more solid at room temperature. However, unsaturated, you're going to have at least one or more double bonds. So here, if we form a double bond between that carbon there and then the rest of our chain, we try to fill in carbons here. This carbon here on the end can form 4 bonds, as always. This carbon here, any carbon can only form 4 bonds, but here we already have 3, so we can only fit 1 more hydrogen. So this carbon has 1 less, so it's not saturated with hydrogen. Same thing for this carbon. This one already has 1, 2, 3. It can only fit 1 more. And then the rest, if it's single bonded, will hold the full amount of hydrogens. There's one there on the end. And another thing about this is anywhere there's a double bond, like right here, between the 2 carbons, a bending occurs in the chain. And that bending of the chain doesn't allow these chains to lay next to each other nice and neatly. So these tend to be oils at room temperature. And so they have what would be a lower melting point. And these, you tend to find these unsaturated types of fatty acid chains in the oils and fats. You tend to find them in plants. So plants tend to provide us oils, and animal tissues tend to give us the fats, which have a higher melting point. There's another concept of trans fats, and these are actually synthetic through a chemical process. And we found out that trans fats are terribly difficult for us to process. We don't have appropriate enzymes to metabolize them correctly, and they can lead to cardiovascular disease. So you're starting to see them removed from food. So here we have 2 different types of triglycerides, and right here is our what was mentioned earlier. This is our glycerol portion. Right here, it started out like this. Glycerol molecule is an alcohol, and the reason it's an alcohol, it looks like a hydrocarbon chain, but the reason it's an alcohol is instead of a hydrogen here, we have hydroxyl groups. So this is the normal glycerol, and a fatty acid chain can be added here, here, and here, and the fatty acid chains are going to have a carboxyl group to them. So generally a fatty acid chain would look like this, and I'm going to draw using a sort of shorthand, so I don't have to draw everything. There's our carboxyl group, and then every carbon after that, everywhere there's a bend, there's a carbon with its full set of hydrogens. So that's what it would look like. So this is a hydrocarbon chain right there, but right there, there's the carboxyl group. So this right here, the carboxyl group is going to interact here with the hydroxyl group as a dehydration reaction, and then we form a bond there, so we can form 3 different ones. So right here we have basically every chain is saturated. There are no double bonds in this area right here, and so this would be a saturated fat. This would be unsaturated, because we see that some of the chains have double bonds. So these would be typical of oils, which are liquid at room temperature, and this would be typical of fats. And on the bottom we have space-filled models that show these types of molecules. Now we have phospholipids. And phospholipids are basically like triglyceride. For the triglyceride, we have the 3-carbon glycerol molecule. And so here is our fatty acid going to it. And a triglyceride would normally have another fatty acid chain there, but if we replace that with a phosphate group of atoms, a molecule of atoms, now we have a phospholipid, and it gets its name because it has a phosphorus there. Now, the interesting thing about this is that the phosphate group is polar, okay, and the tails are nonpolar. So now we have a molecule that has both properties, both polar and nonpolar. And this is kind of typical of our biological membranes. So I'm going to draw a little shorthand here. The way we might be able to draw one of these phospholipids real fast is to put a ball there for the phosphate head, and then just draw two little tails indicating the fatty acids. So we'll see our phospholipids, which are molecules like this, represented in this way. And the thing about these is these guys here form our membranes that we find for the cell membrane around the cell, the plasma membrane, they also call it, and the membranes that make up the little organelles inside. So they do form these biological membranes, the phospholipids. This is an illustration from your book that shows the two fatty acid tails, and they're represented in them differently. Here's just with words. This is the nonpolar part right here, and the polar part of the molecule. So there's your glycerol right there. The glycerol would normally have another, triglyceride would normally have another fatty acid chain there, but instead we're going to replace this with a hydrophilic head there, which likes water, the phosphate grouping there. And then we see here the structural formula to the right, then the space filling model, and then that model I showed you where we just draw the polar head and the fatty acid nonpolar tails there. Maybe I should draw it more, well, let me back up there. Maybe I should draw it a little more right here. There's, I don't even know why I'm drawing it again because it's already there. Anyhow, you get the point. So what does this mean here? This means that that part likes water and this part doesn't. So here's the deal. If I were to get a large number of these molecules, phospholipids, and then just throw them in the water randomly, they're going to form a sort of organized structure. And here's one example where we get what's called micelles. Okay, and the micelles basically are going to arrange themselves where the tails, so here's our water environment all out here. It's polar, and so you're going to have the inside here. The tails are going to move towards the inside, and the polar heads are going to move towards the outside because of the water. So this is what would spontaneously arrange there, and that spontaneous arrangement is called micelles. Okay, and then this is actually how our membranes go, our biological membranes here. It's going to be they're going to form what's called a phospholipid bilayer, and our cells are in water environment. Outside the cell is water, and inside the cell is water. So the only way that these membranes can arrange themselves is going to be where the phosphate heads towards the outside where the water is, and then on the inside, the nonpolar area. So right here is going to be nonpolar. And then out here and here is going to be polar. And what does that do? That provides a sort of a barrier. So let's say we have sodium ion, which loves water. That sodium ion dissolved in the water cannot cross because that sodium ion cannot go through that nonpolar area that's in there. Okay, so it provides a barrier for things that are dissolved in the watery environment, inside or outside. So the only way for these types of molecules to get across is through special protein channels and through other types of proteins that will be covered in a future chapter when we cover membranes there. So that concludes this lecture on the basics of biomolecules.